Good morning, good afternoon, good evening, everybody from wherever you are. We can start the session now. My name is Marla Velasco, and it is a pleasure for me to introduce Riitta Salmelin, our speaker today. She's a professor of imaging neuroscience at the Department of Neuroscience and Biomedical Engineering, Aalto University in Finland. Her research focuses on two complementary lines of investigation: uncovering neuroorganization of human language function by use and development of imaging methods and computational modeling, and examining sensitivity of MEG and fMRI activation and network measurement to different neuro and cognitive processes. She has pioneered the use of MEG in language research and applied multimodal MEG and fMRI and inter-aerial connectivity in the study of human cognition. She is the senior editor of the first handbook on MEG, and associate editor of Human Brain Mapping. Honors include membership of the Academic Europa, while a Young Investigator Award by the Organization for Human Brain Mapping, and the Justine and Eve Sargent Award. It is a pleasure for me. And as the other plenary talks, we are going to present the pre-recorded video, and afterwards she will be able to answer your questions and answers, and we'll answer everything live. And so please, during the presentation, post your questions on chat, and after the video finishes, she will be able to answer all of them. So we can start.
I would first like to thank the organizers for giving me the opportunity to share my views on what neuroimaging can tell about brain function and how machine learning and deep neural networks may help us in that endeavor. In the brain, neuropopulations are firing all the time. We would like to know, be able to track them and know what their functions are. Neuroimaging and neurostimulation provide multiple windows to brain function. The oldest one of them is electroencephalography, or EEG. And the new ones include functional magnetic resonance imaging, magnetoencephalography, positron emission tomography, near-infrared spectroscopy, transcranial magnetic stimulation. In this talk, I will show data collected using fMRI and MEG. But then we also need to ask, what kind of windows do fMRI and MEG then provide to us to brain function? And for that, we should take a closer look at a small region of the cortex. And what we would see there would be large populations of pyramidal cells with the apical dendrites nicely aligned. And here's a schematic view of such a setting. With we have a schematic view of a population of pyramidal cells, and then there's a nearby blood vessel with the red cells. And so if there's a neurofiring in large population of neurons simultaneously, then MEG can see this activation as a change of magnetic field in real time. Now these neurons need energy, so they suck oxygen from the blood flow, and thereafter there's a rush of blood flow to that region, which is something that fMRI can detect, but this is a very slow phenomenon over a period of seconds. So fMRI is limited by that. In MEG, we have very high time resolution in real time, but not so great spatial resolution, whereas with fMRI, we can get excellent, really great spatial resolution, but the time resolution is limited. So they're always complementary measures, and they always see part of what's going on. We can think of this, in some ways, this neuroimaging signals as almost like a blood sample. So we just get these measures, and then we can extract multiple metrics from those signals. So we can look at local effects, changes of activation, and network effects, connectivity, that type of effects. But even when we have these neuroimaging-derived effects, we can measure them, we will need some sort of models to interpret their functional roles. We do not have yet a model of actual, like model of the brain itself as such. So mostly our models are domain specific. And so we're thinking of what kind of processes might be needed in such and such tasks and how might they be mapped onto what happens in the brain. And the most basic ones are those which are based on, one usually starts from is acquired or developmental disorders in specific aspects of sensory, motor, or cognitive processing. And then when some part of that process is broken down and other parts seem to be functioning normally or less influenced, so then we conclude that there is a kind of probably a separate stage of processing, a modular structure. And then the result is kind of this descriptive modular models of processing stages. But we really need to have quantitative models, and that's what people are trying to develop, behavior-based quantitative models, because we do get these quantitative measures from neuroimaging metrics. An even more advanced version, which is something that people are more recently working more on, are computational models of representations and mechanisms. Because we would really like to find ways of having models of the computations that the brain signals reflect, so that we would actually better understand the brain signals and how the brain works. And here I hope to illustrate some of these different steps and advances using language function as the example. The starting point for any new tool one gets, like neuroimaging tools, is that one wants to get to the point where one can show that there are reproducible group level descriptions of processing stages. And this is something that the neuroimaging community has really focused on obviously for the first few decades. But now and this is something we think that we can then start building on. And now I'll show you some examples of what we think we know about, for example, language perception. So when we think of a simple thing, presumably simple thing like word reading, here is a pretty basic behavior-based descriptive model. So when we see a word swan, visual features must be processed first before the answer proceeds to the content. Apparently first the level of single letters and as a whole word, which further activates the word's meaning and possibly also its sound form. And the meaning is accessed by about 200 milliseconds after the word has been shown on the screen. Well, in the brain, what it looks like is we can take a look at the dynamics over the first 650 milliseconds after word onset. This is one individual left and right hemisphere. And we will see that activation starts at the back of the head in the visual areas, in this subject particularly on the right, and touch on the left, again the right hemisphere. And then by about 400 milliseconds, we see this strong sustained activation here in the left superior temporal cortex. So plenty of activation in both hemispheres, but what are the functional roles of these signals? Now, using lots of experimental paradigms, different stimuli, different tasks, it seems that at least in the left hemisphere, there are some activations for which there seems to be quite salient functional roles in word reading. And now here the curves, they show the time course of activation in this cortical area, starting from time 0 when the word is presented on the screen until 800 milliseconds after it. So first we see here at the back of the brain, in the visual areas, early activation at about 100 milliseconds after stimulus onset, which is still non-specific. It doesn't care whether the stimuli really are letter strings, words, whatever. It's just something visual. But then just only 50 milliseconds later, at about 150 milliseconds after stimulus onset, in the left inferior temporal cortex, there's activation that seems to really care about letter strings. However, it doesn't care whether they are words or non-words. It's just that it looks like a letter string. But then subsequently, from about 200 to 600 milliseconds, in the left superior temporal cortex, there's activation that does differentiate between words and non-words. And it also seems to be reflecting not just processing of meaning, but also processing of sound form, syntax. So how do we come to this kind of interpretations? I'll use cortical processing of meaning as an example to illustrate one way of getting to this timing or processing of meaning. So here you will see sentences. Please read them. In reality, these sentences were finished, so all the word lengths and whatnot were matched, of course. But you may have noticed that there were different types of sentences. And now the signals were averaged across, we had 1 trials per sentence type in order to get a good signal-noise ratio. So the problem is the brain is doing lots of things all the time. And when we want to extract something specifically related to the task we are implementing, we often need to average quite a bit to be able to get rid of this unrelated noise from our point of view. And when we do that, we see that for the sentence types where we have the expected ending, like the piano was out of tune, everybody knew it's going to be tune. So then what we see in the brain in the left superior temporal cortex, a very, very flat response, almost nothing. However, as soon as there's the totally wrong ending, which you may have noticed, the pizza was taught to sing, there's a huge response in the brain, like really processing it. And also when there was an ending of the type, the gambler had a streak of bad luggage instead of luck, where the word is wrong, but it shares its initial letters with the expected word. So then, interestingly, we see a strong response, just like for the wrong words, but there's a slight shift forward in time because the reader is a little bit fooled by these initial correct-looking letters. Another critical condition is this condition, like when the power went out, the house became quiet, when most people would feel in dark. So the ending is plausible. It's unexpected, but it's plausible, semantically plausible. And the response falls nicely between the wrong and expected. So we have this very nice graded response. And we see a very similar graded response for when the subject listens to these sentences instead of reading. So this type of graded response, similarly for auditory and visual visual stimulation, points to processing of word meaning regardless of the input route. Now, of course, when we listen to words or read them, the first initial processing stages must be different. And that is indeed the case. So also the model, again, this kind of basic behavior-based descriptive model of spoken word perception says that when we hear a word swan, first, of course, the speech enters the ear as sound waves from which the brain somehow magically extracts speech sounds. And we'll get back to this point later. And speech sound sequences which further activate the word's meaning. And the meaning is accessed again by about 200-300 milliseconds after stimulus onset. In the level of the brain, what it looks like is that there's activation all the time, more or less, here in the superior temporal cortex. But there's first a response at about 100 milliseconds, which is just to any kind of sound. And then if the stimulus is a word, there's this type of sustained response, which reaches a max about 300-400 milliseconds after stimulus onset. And although the activation stays pretty much in the same area in the brain, there are different functions reflect at different times in this response. So by 100 milliseconds, the signal shows sensitivity to speech-specific acoustics. By 150 milliseconds, the superior temporal cortex can differentiate between speech sound categories, so different phonemes. And from about 200 milliseconds onwards, the sensitivity to word meaning, as we already saw. However, then one of course should ask, so what happens? Can we say what happens when there's a disorder of language perception, like in dyslexia. So what does the brain do? So in dyslexia, there are difficulties in learning to read and write despite normal intelligence and social cultural opportunity. Problems are generally reported also in phonological processing, and dyslexia is typically discovered during the first year at school. And now we ask, what does it look like then in the brain responses? And here, this slide again recaptures the findings that I showed you earlier on fluent readers. So we have in word reading, letter string processing, meaning processing, in spoken word perception, this sequence from activation of the superior cortex. And now here is data from adult dyslexics. And what we see that there's a striking difference in word reading, in letter string processing, which is very heavily impaired in dyslexia. And then the subsequent activation reflecting processing of meaning is heavily delayed by about 100 milliseconds. In spoken word perception, again, there are differences in the signal strength already at 100 milliseconds. And again, by the time we get to semantic processing, there's a delay, now about 50 milliseconds, so a little less than in reading. So what we see in both modalities is that problems the differences from the normal pattern start to appear at the earliest language sensitive processing stages, and by the time we get to processing a meaning, there's a delay. And this delay is particularly emphasized in reading. Now, of course, now you saw rather simple experimental designs, and this is often what we use, rather simply, very heavily simplified experimental designs, mainly because that's how we can really control the stimuli and the tasks and what the subject does, and this helps then the interpretation. But in reality, in natural language processing and in any kind of natural cognitive task, of course, it's very likely that what we actually do in what our brains does is partly different from what we see in these rather simplistic tasks. And indeed, here's an example from experiment where subjects listened to their own speech, which had been recorded earlier, and another person's speech. Also, amplitude modulated noise, which was had the same envelope as speech, compared then to speech signals. And the stimuli had been produced at slow, normal, and fast speed rates. And what we saw in the brain was that the effect of rate showed in the left hemisphere, where perhaps we might have expected these effects, and also so that the neural that there was stronger activation, presumably stronger neural effort, as soon as the rate deviated from the normal rate. So whether it's slower or faster. But now what we saw was that in the right hemisphere, there was very much activation, and that's where we saw the effects of linguistic content and of the speech ownership. And this is an interesting area, this right temporoparietal junction, because it has been previously, in other studies, has been linked with attention and social cognition. And this is quite an interesting thing, because it kind of suggests that in natural language, there's this, when it's communicative, natural language, there's a very strong social attention influence that it has. So we see much more right hemisphere activation. And this often is in general, when we get to this more naturalistic language tasks, it seems that we are getting more right hemisphere involvement as well. So things may be different then. But this was kind of like, basic stuff that we have been doing and trying to build upon, doing in a way kind of traditional approaches in doing language studies. But then now we would like to move towards neural level representations and mechanisms. We would like to understand what kind of what is there. So, for example, when we just thought about semantic processing, like this activation reflects semantic processing. But then we get to the question about what are the cortical representations of item meanings? How do we know that different items mean different things? How does it show in the brain? So, for example, we also have this feeling that that there's a certain structure to things, like that dog, horse, and cat, they are all household animals. They are linked, they go together. Boat, train, car, they are vehicles. Drill, saw, scissors, they are tools we take in our hands to get stuff done. So how do our brains carry information about the qualities that form the item meanings and their relationships? Now, language technology offers interesting ways to model semantic relationships quantitatively by the context in which words appear in large text collections. Tools like methods like Word2Vec, for example. Now, when we have a text-based model, we have brain data and we have machine learning. We can try to learn the relationships so that in the end we would have a brain representations of the model features, and hopefully we could then take just any item and predict its brain patterns, or try to decode from the brain what that item, or at least at this point, just what the item is. And this is our hope, and we can do it to some degree. And this is a line of research that was studied by Tom Mitchell in Carnegie Mellon, where he had many different items as stimuli, showed them to subjects, measured their fMRI patterns, and then he had this feature space in between, where he made an educated guess of something that might at least approximate the way that information is organized in the brain. And he chose these text-based semantic features, like, for example, like Word2Vec. And that the way we use language, to use that as a model of maybe how the brain is also, not unreasonably, model that might be a good model of how the brain also does it. And that's what he did, and trained the model, and was able to eventually get to a point where he could predict for airplane that this is what airplane should look like in the brain, and decode to a degree from the brain that what was there probably was airplane. And the important thing in this approach is that it generalizes to unseen items. There are also other approaches that have been that are used to try to link the brain, represent what is brain signals to something that could be semantics in the kind of semantic model. And so here's an approach by Clark and Tyler, where they used representational similarity analysis as a method, where they used various types of, they showed the subjects many different items, and they used various types of features to build distance matrices among these items. And then they measured fMRI signals to each item, and then sought to link the two. So here we have distance matrices between the items, so items on the horizontal axis, on the vertical axis, very low level visual model, then models of semantic features, and then they also model different kind of make models of just certain categories, semantic categories like animals, tools, musical instruments. And then in the brain, what they did was they went through small region one after the other, and took the voxels in that region and saw whether the voxel pattern in that region, whether the distances between the voxel patterns formed this type of matrices that would correlate with one or the other of the models. And what they found was that particularly at the back of the brain, the visual areas, there was a good correlation with the kind of visual model, whereas the semantic features, that kind of model linked much better with activation patterns more anteriorly along the ventral stream. So these are kind of probably most frequently used methods. But the question is, so this is a very good approach, use something like Word2Vec or whatever, but what does it, how true is it? Does it actually reflect something that the brain does? And one important check is whether it's not just concrete words, but also abstract words, semantics that can be modeled. Because abstract words, they don't have any sensory you don't have any sensory experience about them. It's something that they in a way exist in language. So can we still model them? And this study looked at that. So there were a large number of Finnish words, and then models, word-to-vec models built from a large text corpus. And there were items which were names of animals, body parts, buildings, human characters. It's like priest, the king, teacher type of things. Nature, objects, artifacts, medium abstract words and highly abstract words. And it turned out that it was possible to use word-to-vec to text-based model to decode accuracy, to decode these items from both concrete and abstract items from MEG data with an accuracy of about 65%, which was significant. So it seems to work, but in order to interpret what it means, we also had a separate model, which was just asking people with a questionnaire to tell how abstract they felt the words were. So there's now a separate model. And we asked for each of them using representational similarity analysis where these models work in the brain. And it turned out that there was a great overlap, these yellow areas, between the statistical model based on the corpus data and this questionnaire-based abstractness model. And this overlap now then suggests that this word abstractness seems to be an emergent property of that arises from the way we use language. In the statistical regularities of our language use suffices to account for at least the neural level, large part of the neural level effects, especially in the left hemisphere, about the of abstractness, of the word abstractness, which is kind of nice. Also, another further question of whether these models work is whether we can model some words which were never actually shown, that were only conjured in one's mind. And here we used a childhood guessing game type of design, where such were shown clues: has legs, has a thick skin, has a trunk, and they guessed elephant. Is a fruit, is peeled, monkeys eat it, banana. And now the question is, when the three clues have been presented, which actually don't contain anything about elephant or banana, so at this point, what do we see in the brain? Is it does the brain activation reflect the target word, so elephant or banana, the three clues, so the words which are in the clues, or all kinds of features that are related that we generally in pre-production, if we are asked to describe an elephant or banana, all those features that we tend to generate to those target words beyond the clues, which this very limited set of clues that were provided in the experiment. And here is a semantic space banned by the word-to-vec features, but we have now the only three-dimensional the visualization, where the orange dot represents the sum average of the three clues. Blue is the target words semantic coordinate, and green is the semantic coordinate for the for all features related to sum of all features related to the word beyond the clues. And the way we did this is that we trained a model using all items except two, and then we tested with these two left out items of how the thing worked. And here we have the fMRI voxel values on the horizontal axis on the rows, and we have all the trials, so the different items on the rows on the vertical axis, and here and on the right we have the semantic coordinates estimated with word-to-vec for all the items, for all the trials. And now we learn the relationship. We learn what are the fMRI patterns of the different semantic coordinates. Then we take the left out items, and then we take the fMRI values, the voxel values. We see what are their predicted semantic coordinates, and we check whether they correctly, whether they are classified correctly to the original semantic coordinates. And of course, we do all the permutations. And now the part where we test the different models is this. So how we come to these what is the different model of what we are testing for the brain activity? What we find out consistently across subjects is that the accuracy, prediction accuracy, increases from just using the third clue to all the three clues or the target word. But importantly, there's a significant improvement still for including all the features that we tend to assign to those items in pre-production. So what this suggests is that we see semantic completion from bits of information. We are given something, but our brain expands the view. So these approaches seem to work pretty well, but we can also use these similar ideas to look at other things than just semantic processing. We can really try to look into mechanisms. And now we'll get back to this spoken word perception and this question of how we magically, from sound waves, something becomes speech sounds. And this is really an age-old question of whether there are different mechanisms somehow for processing speech versus other natural sounds. Is there something special about speech? And here, if we go back to saying that if we have a spoken word like huilu, so that's flute in Finnish, then we would have an MEG response over the auditory cortex, something like this kind. And now we can have various models, acoustic models for the spoken words. The simplest one is the simplest description of acoustic features of sounds is a Fourier transform, like a really simple one, no time information, just a frequency. There are more advanced versions like modulation transfer function, which also includes descriptions of spectral scales and temporal rates. It's still a stationary model. So if we have something like an MEG signal which has time information, we'd like to use it, we can't, but we would just have to lump everything together. And we really would like to make use of the time information. And the simplest time-sensitive model, which is still computationally feasible as well, with all the data that one can get from MEG, is a spectrogram. And we developed a dynamic model which actually tries to reconstruct each time frequency point in the spectrogram from MEG signals with a specific lag, taking into account the fact that we only hear the sound and the MEG response comes later because there's a delay from the auditory signal to the cortex. We take that into account, and we have this kind of dynamic model where we have a lag which from the unfolding stimulus sound to what the MEG sees, and try to retrace what the spectrogram would look like. And now with these different models, we modeled spoken words and corresponding environmental sounds. So we also, if we had a spoken word like huilu in the stimulus set, we also had an environmental sound which was a flute melody as a stimulus. So they would have the same semantic endpoint. And we would have very similar activations. Otherwise, overall, when you look at the signal patterns, they're just very similar. But it turned out that the models work differently, so that for environmental sounds, it didn't make a difference whether one used a stationary or time-sensitive model. However, for speech sounds, the spoken words, it made a huge difference, so that there was a huge increase in prediction accuracy by using a time-sensitive model. That clearly worked so much better. And it was best at a lag of about 150 milliseconds between the unfolding sound and the cortical response. And so it really seems that there's something special in the way that the brain chooses to process speech, presumably to have perform an instantaneous acoustic to linguistic transformation, because this is a bit more advanced. We just can't kind of take it as okay, this is all the parts there are, but you really have to online transform these acoustic signals to something more abstract. finally, it would be really great to move to forward to actual computational models, full computational models of language. For example, in this case, word reading. So I'll just show you again the sequence of from these processes, which we think are processing of visual features, processing letter string, processing of meaning, and show what there is. So these dots here, the colored dots, show responses of different individuals and different trials for words, and then pseudo words, so meaningless word-like strings, consonant strings in green, symbol strings in red, and words, real words embedded in noise, so that it's really hard to see them, in visual noise, so that it's really hard to see them in this purple color. And we can see that for visual features, this is exactly kind of a definition of it, that there's everything is the same except for when you have this really strong visual noise, which is then requiring much more processing. For letter strings, all the words, pseudo words, consonant strings kind of go together, and then symbol strings are separate, and then also words noise because it's hard to see them. For processing of meaning, words and potentially meaningful pseudo words, they go together, and the others have much lower signal. So this is what we see in the brain. And now Marijn van Vliet constructed a convolutional neural network of this type, starting from what is already working fine for visual object recognition. He took the ideas from there and started transforming it into something that would work for words. So he had used a large number of 10,000 words when there are only about 4,000 units, so that there couldn't be just a simple word per unit correspondence, but the model would actually have to do some sub-lexical representations. And it worked fine. In the end, at least it was able to, when you see puku, it was saying, yes, there's puku. And this is what the activations at these different nodes look like. So the first ones, they certainly look a lot like the visual feature processing, especially the first three. They have very similar patterns, just the words in noise is different. But then at levels 6 and 7, we start to see something which looks very similar to what we consider string processing, so that really only the symbol strings and the words in noise tend to behave differently. And then finally, this final stage, it looks a lot like, has a very similar pattern to this lexical semantic processing stage. So this is, of course, only the beginning, but I'm really impressed. And I was really happy that this kind of results could be obtained. And I'm really looking forward to seeing how this type of models may help us to really better disentangle what happens in language disorders, in development. What are the transitions and computations that are actually reflected in these? How does the transition happen, for example, between the visual features, the strings? So the starting point it's not huge yet, but the idea, I think, is really important. And finally, I want to touch upon something which I think is increasingly important, is to acknowledge individual variability in brain signals. And let's go still back to this reading example, where we have this semantic processing, which is a group result. These are the data from individual participants. So what we can see is that there is certainly a very similar effect in everyone. We can see that, but the actual timing and the exact kind of relationship between signal strengths, they are quite variable between individuals. And it's certainly very likely that also this, not just semantic processing, something that we call semantic processing, and these meaning representations, they are also likely to show individual variation. And just to illustrate that point, here's a small child seeing lovely red berries and wants to taste them. And the reaction is like, oh, awful. This is really I shouldn't have eaten that. And what this little child, who actually is me at 2 years, didn't know is that 11% of Finns have a specific variant of a certain gene that makes lingam berries taste particularly or even unbearably bitter. And it's for sure that this type of experiences will influence the way that me or someone else will think of a small red berries after that. So it certainly will change, will have an influence of meaning representations. And I think we may assume that individual differences in meaning representations can be shaped by nature and nurture. And what is really lovely is that hopefully now by this using machine learning, text-based semantic models, other semantic models, whatever we can think of, may help us to actually start digging deeper into these questions as well. And here's just a small example along those lines, where the study asked how much data the brain needs to accumulate to reach item meanings. And here's the idea was that subjects were shown this item, naturalistic images of items. So we had MEG data for those items, and we had the word-to-vec semantic vectors estimated for those items. But now what we tested with the model was that we increased systematically the time of the MEG data that we allowed the model to know about. And we always left out one item at a time. And then for that one item, we then, using the regression model learned through using the other items, we computed a predicted semantic vector for that semantic coordinate for that item, and asked how close it came to its actual target vector. So how close to the meaning had we come already? And what happened was that we were able to see that there was this kind of, when we increased the time of how much data we gave to the model, there was finally a plateau reached, after which, when more data was added, there was no improvement it didn't get any closer to the actual meaning. So then we say that this is a time which we think is a time when one reaches a meaning. And this time varied quite dramatically between individuals. So they had their own plateaus, own shapes, and the plateaus between individuals varied between 420 to 640 milliseconds, which is, in the brain terms, it's really 200 milliseconds is a really it's a lot of time. So we are only starting to dig into the neurobiological basis of this type of variation. And at least one obvious thing to ask, one obvious question to ask is, how individually unique is our global brain function? And if we think of just recording data from one subject, this is 8 seconds of data from an individual recorded over the left and right sensory motor areas, posterior visual areas. And when the subject was just resting, eyes closed. And on the right, we see a typical frequency spectra for a healthy individual. So the back of the brain, we will see a very strong 10 Hertz peak. And over the sensory motor area, small 10 and 20 Hertz activity. And it turns out, intriguingly, that these individual spatial spectral patterns can be distinguished remarkably accurately. We used Bayesian reduced rank regression on a data set of 100 sibling pairs, so 100 families, 200 subjects. And we computed just this simple spectra, spatial, just a power spectra. And we aimed to train the model so that we tried to find spectral compositions that would maximally differentiate between the phenotypes, so between the families. And we used always 90% of the families in training, 10% for siblings testing. And on the right, we see a coordinate system spanned by 2 of such spatial spectral components. And each dot shows one subject. So we see that the same individual, for 2 different measurements of the same individual, they really go together. And siblings are also quite close to each other. And it turned out that individuals could be recognized with very high accuracy across different tasks and recording sessions from only a few seconds of data. Now, what this means, we don't know yet. This is really interesting, extremely interesting, could have huge importance for studies on healthy subjects, neurological patients. But now we are at the stage where we try to understand what this means and what does this individual variability link? How does it link? How does it link to individual variability seen in task conditions, for example? So now to conclude, and I hope I have managed to somehow convey these ideas to you during this talk. I think that corpus-based and neural network computational models hold great promise for interpreting cognition-related neural effects. However, a thorough understanding of both models and neuroimaging signals is essential for informative functional interpretations. And I think sometimes maybe we don't value it enough that one really has to, in a way, have a command of two areas or multiple fields to appreciate all those little things and little problems which become big problems in interpretation if we don't really understand them. And I think individual variation must be fully appreciated in next generation human neuroscience. It's something we can't overlook anymore. And I very much hope that computation and believe that computational models may help to determine the processual and neurobiological underpinnings of such individual variation. Finally, I would like to thank you all for your attention and obviously thank all my wonderful collaborators in Aalto University, elsewhere, who have really made huge contributions to the work I presented. Thank you.
Thank you very much, Riitta, for this very interesting talk. And now I open the floor for questions. I have one here from the chat. I'm going to read it for you. In the context of the environmental sound versus speech perception experiment, would it be possible that motor function, vocalization, is what makes the difference?
Thank you for the question. Yes, that's a good question, but I think it's a larger question than I'm not sure if this is what was intended, but what I can say simply about that particular data set is that it isn't enough, at least, that it's something that people can produce themselves, because there was also a condition where it wasn't speech, but it were natural sounds that people produce, like laughter and coughing and stuff like that, which are also produced by the same vocalization system. And then that operated like an environmental sound that didn't follow anything in time. But of course, there's a bigger question. I think this is an absolutely interesting and correct point, that there's a larger question of how, in general, we build our representations of phonemes. That surely we learn to listen clearly before we really learn to produce very well. But it's for sure that, and there's pretty much evidence that it's very likely that we keep polishing and finalizing our perceptual representations, our phoneme representations, when we learn to speak, because they're kind of interacting. So whatever the representation is in the end, it's most probably influenced also by the way we produce. But in this particular task, the people were not vocalizing themselves. So if they used those representations, they used it because they had already built a link between them, basically. So that's how I would answer to this one. But no, it's not like an obvious thing that it would be that.
There's another one here. To what extent are the results helping to challenging existing theories of language and show perhaps new possible NLP concepts?
Yeah. Yes, well, obviously, I'm hoping that the more we learn to link what we see in the brain to at least some models, then the more we can also feed back to those models, hopefully. But I think we have to accept, and I'm not saying that this wouldn't be possible. I hope it will be. But the problem is partly that what I said at some point was that we don't really have a model of the brain itself. So in a way, we have balls in the air, and they keep moving when another ball moves, and we don't really we don't have a full kind of, yes, they are in this part of that picture, that where that ball should be. So I think this is one of the things we have to live with for now, that we keep developing one thing, and then we kind of have to accept that we don't have a real fixed point. We have some fixed points, which in the end is our behavior observations. So yes, at this point, I would say we are not at that point when we are necessarily suggesting that there would be new NLP concepts. But I would think, and I expect that that would be possible, because it has been possible to brain data has started. It didn't do it in the beginning, but it has already in between kind of influenced those low level, let's say, the first earliest descriptive models of language, for example. They have already influenced, but it took years. It took like 10 or 20 years before you start to say that, well, actually the brain seems not to be doing that. And I think that's probably the same thing is going to happen with some of these, say, statistical models of language NLP concepts. So I'm hopeful, but not at this point, I think, yet.
It will take some time, right?
It will take some time, yes.
Another one, it's from Yonsuk. He asked about the context of environmental sound, and he's saying that, yes, indeed, it was more generally curious how much motor function factors into all of this. Thanks. It
does factor in, yes. But it's not fully understood exactly how they factor in, but yes. Yeah.
Well,
anything else?
Anybody wants to ask another question? We still have some time.
Feel free to send me email afterwards if you think
of course,
something.
yes, certainly. So, well, I want to, there is one. In the case of dyslexia and other conditions, are ideas coming to better help individuals? But this should include perhaps all of us, as we might all better improve ourselves. I think there are perceptual examples of this.
Okay, so yeah. So yes, I think dyslexia in a sense is a good example of what can be done. So so I think that in the in the early days, it was more like, there was a time when I started working on dyslexia where there was this feeling that, dyslexics felt often, apparently, that, not surprisingly, felt that somehow people thought they are stupid. And which is not, it really was clear already at that point that no, it is not a question of intelligence, but it was more like this, like if you can't read properly, it's something about you don't try hard enough or something. And then I remember how many participants, when they joined in, they said, I'm so happy to hear that there's something in my brain, the specific thing in my brain. And in a way, this thing that it's something that just doesn't develop properly when you learn to read, something that in a normal reader becomes kind of active. Like we are very good at recognizing faces. We have a very similar early activation to faces. And then it seems that letter strings as a visual experiment, it's a visual stimulus. It's so important that our brain kind of develops this kind of fast route for processing letter strings, which makes sense. And this probably makes most of us like really fluent readers, because it's such an automatic process. But it seems that this automatic process, it's not functioning properly in dyslexics. And of course, that kind of then pointed even more, that kind of results, which came from different directions, pointed even more to this importance of really putting the effort in training to people, to children, trying to identify children at risk for dyslexia early enough, when it's still possible to influence, but let's say whether it might be possible to influence this process, because then it seems that when you are after a certain point, it's very hard you can't develop this letter string response anymore in an adult, apparently, it seems so. So it really kind of still brought it even more to the point of before school, and emphasized this importance of finding tools to identify such individuals. So I think all this kind of work together kind of gives you hints of when something should be influenced. And then in principle, of course, it gives you also measures of whether something worked or not. Of course, mostly in case of like dyslexia or something, you don't need to go to the brain to check whether it worked. You want to see if it starts to work. But the importance is that you should be able to find ways of identifying these things early. And kind of this kind of work may give you idea about time windows when you should be working on, and maybe like highlighting the particularly affected parts of your system and stuff like that. So yeah, that's, I guess, what I would say. It has helped already, I think. Yeah.
Another one, at present, the working mechanism of the brain is based on observation. Is there other methods, for instance, deductive methods to study the working mechanism of the brain? Deductive.
Yeah. Well, yeah, I think it's still mostly an observation.
Yeah.
Primarily, mainly also because I would like to say that it's also partly because we are not quite sure even at this point what it is that we should be observing that would be most informative of what the brain does. Like I said, with imaging or something which we can use in healthy individuals, we can't really go into the brain. We kind of also there we would need to understand what that part we can see actually reflects, and maybe that it may still be that you are using the metrics which we think are informative, but it might be that it's actually some other metrics. If we understood the thing better, we might be digging out some smaller metrics and more kind of subtle metrics, which would be actually be more informative. So I think we are still kind of also at the same time while we try to do clever things, we should keep our eyes open for other things we should be looking at.
Yeah, as I said, there is no model, and there are lots of things that we still need to learn and understand, right? So
so you try to do more, but you have to keep your eyes open also for other things.
Thank you, Riitta, again. There are some other questions. I think it's the last one because we need to close the session.
Yeah.
But do you have any comments about schizophrenia?
No.
I'm thinking the gamma rhythm disruption.
Yeah. Schizophrenia, very tricky, has remained very tricky also in trying to understand what would be a good measure. I know that there are people loving gamma band, and then there are people thinking that, oh, there are also other things in the brain which are doing interesting things. I think I have to say that gamma itself as alone is no solution, I think, to anything at this point. It's an interesting phenomenon in the brain, and in many ways, I think now there's a very common way of thinking that, well, network disruption is a problem of everything, for everything and explaining everything. Of course, again, we will find out that it's not explaining everything. But yes, gamma band is one of those things. Okay. There is, I was thinking whether you referred to, now I see that there's an answer to this, the deductive. Yes, TMS, absolutely. So if you want that, I agree. I was thinking a bit differently about this is the deductive part, but I agree that stimulation does something, and there are many ideas about like stimulation and then measuring. The problem, of course, with TMS is the same as in a way as with imaging, that it's hard to know what are all the things you are stimulating. But stimulation and imaging are both very valuable methods. But one has to know how to use them.
Of course. Thank you very much again for a fantastic talk. And thank you all that attended. So we are closing the session now because we have other sessions in the
Thank you
to start. Thank you very much. Bye-bye, everybody.
Bye-bye.